Hi, I'm Melinda Rose.
And I'm Laura Marshall, and this is Light Matters for June 15th, 2011. In this week's show, a kidney cell becomes a living laser, rods are directly imaged in a live eye for the first time, 1-atom-thick optical devices are proposed, and the historic two-slit interferometer experiment gets a modern makeover.
A human embryonic kidney cell, genetically engineered to include a light-emitting jellyfish protein, forms a living laser capable of producing visible nanosecond pulses of light. Instead of using traditional synthetic gain medium materials, researchers at Massachusetts General Hospital used green fluorescent protein, or GFP. The irregular interval structure of GFP-expressing cell causes the apparently random pattern of laser emission. The project was motivated by basic scientific curiosity by wondering if there was a fundamental reason why laser light doesn't appear to occur in nature, and if they could also achieve biolasing, the scientists said. GFP was chosen because it can be induced to emit light without the application of additional enzymes. Its properties are well understood, and there are established techniques to genetically program many organisms to express GFP. To determine the protein's potential for generating laser light, the researchers first assembled a device consisting of an inch-long cylinder with mirrors at each end, filled with a solution of GFP and water. After first confirming that the solution could amplify input energy into brief pulses of laser light, they estimated the concentration of GFP required to produce the laser effect. The cellular laser was assembled by placing a single GFP-expressing cell with a diameter of less than 20 microns in a microcavity consisting of two highly reflective mirrors spaced 20 microns apart. Not only did the cell-based device produce pulses of laser light, the spherical shape of the cell itself acted as a lens, refocusing the light and inducing light emission at lower energy levels than required by the solution-based device. The cells used in the device survived the lasing process and continued producing hundreds of pulses of laser light. The ability to generate laser light in vivo could be useful for photodynamic therapies or novel forms of imaging. One of their long-term goals is finding ways to bring optical communications and computing into the realm of biotechnology. They also hope to be able to implant a structure equivalent to the mirrored chamber right into the cell, which would be the next milestone in their research. The work appears online this week in Nature Photonics.
The tiny light-sensing cells known as rods have been clearly and directly imaged in the living eye for the first time thanks to an innovation in adaptive optics. This will help doctors diagnose degenerative eye disorders sooner, leading to quicker intervention and more effective treatments. Early detection is critical in the treatment of eye diseases, and being able to see the cells you're trying to rescue is a critical first step in the process of restoring sight. One of the major hurdles in detecting retinal disease is that by the time it's detected, significant cellular damage has taken place. Diseases amenable to early treatment often affect the numerous and very light-sensitive rods of the eye. The problem is, light passing through the front part of the eye gets bent and distorted, which doesn't affect vision but poses a significant barrier to medical imaging. The solution involved using adaptive optics, which in astronomy corrects for the blurring effect of Earth's atmosphere, effectively removing the twinkling from starlights and rendering cosmic objects as very sharp points of light. Adaptive optics technology was first used to study the inside of the eye in 1997. In that system, called an adaptive optics ophthalmoscope, a laser creates a reference point that is used to correct the blurring of the image obtained with a fundus camera. Today, the fundus camera is commonly replaced by a second laser for imaging. In their improved design, the team was able to push the device's resolution to its optical limits of nearly 2 microns, or the approximate diameter of a single rod in the human eye. They accomplished this by simply folding the spherical mirrors that act as lenses in the instrument into a 3D structure. By doing that, the image quality of the retina was improved sufficiently to clearly resolve the contiguous rod mosaic, as well as the entire cone mosaic at the foveal center. With the improved design, even the smallest cone cells at the center of the retina, known as the foveal center, can be seen very clearly. By moving the laser point across the retina and correcting the distortion along the way, an accurate image emerges line by line in much the same way that a CRT monitor renders an image. The researchers' next step is to develop a clinical model. The work is described in the OSA journal Biomedical Optics Express.
1-atom-thick metamaterials can be achieved by controlling the conductivity of sheets of graphene, University of Pennsylvania engineers theorize. The study of metamaterials has grown considerably in recent years. It is based on the idea that materials can be designed so that their overall wave qualities rely not only upon the material they are made of, but also on the pattern, shape, and size of irregularities known as inclusions or metamolecules that are embedded within host media. These unusual properties generally have to do with manipulating electromagnetic or acoustic waves. In this case, it is EM waves in the infrared spectrum. Changing the shape, speed, and direction of these kind of waves is a subfield of metamaterials known as transformation optics and may find applications in everything from telecommunications to imaging to signal processing. The research shows how transformation optics might be achieved using graphene, a lattice of carbon a single atom thick that has unprecedented conductivity and possible applications in electronics. Graphene also has the ability to transport and guide EM waves in addition to electrical charges, and its conductivity can be easily altered. In this marriage between graphene and metamaterials, the different regions of conductivity on the two-dimensional sheet function as the physical inclusions in three-dimensional versions. The examples demonstrated with computer models include a sheet of graphene with two areas that have different conductivities, one that can support a wave and one that cannot. The boundary between the two areas acts as a wall, capable of reflecting a guided EM wave on the graphene much as one would in 3D space. Another example involves three regions, one that can support a wave surrounded by two that cannot. This produces a waveguide which functions like a one-atom thick fiber optic cable. A third example builds on the waveguide, adding another non-supporting region to split the waveguide into two. Other applications include lensing and the ability to do flatland Fourier transforms, a fundamental aspect of signal processing that is found in nearly every piece of technology, audio or visual components. The work will pave the way to the thinnest imaginable optical devices, the researchers said, because you can't have anything thinner than one atom.
By applying a modern measurement technique to the historic two-slit interferometer experiment, scientists soon may be able to measure reality without distorting it. Quantum mechanics tells us that a tree falling in a forest with no one there doesn't make a sound. It also states that anyone who does happen to be listening interferes with and changes the tree. In the historic two-slit interferometer experiment, a beam of light shining through two slits results in an interference pattern on a screen behind. That seemed to establish that you could not watch a particle go through one of two slits without destroying the interference effect. You had to choose which phenomenon to look for. In their modern experiment, the researchers succeeded for the first time in experimentally reconstructing full trajectories that provide a description of how light particles move through the two slits and form an interference pattern. Howard Wiseman of Griffith University proposed that it might be possible to measure the direction in which a photon was moving, depending on where it was found. By combining the information about the photon's direction at many different points, scientists could construct its entire flow pattern. Their experiment used a new single photon source that was developed at the National Institute of Standards and Technology to send photons one by one into an interferometer that was constructed in Toronto. Using quartz calcite to measure the direction as a function of position, they discovered that their measured trajectories were consistent with Wiseman's prediction. The original double-slit experiment played a central role in the early development of quantum mechanics, leading directly to Niels Bohr's formulation of the principle of complementarity, which states that observing particle-like or wave-like behavior in the double-slit experiment depends on the type of measurement made. The system cannot behave as both a particle and a wave simultaneously. The recent experiment, however, suggests that this doesn't have to be the case. The system can indeed behave as both. The work is expected to contribute to the ongoing debates over the various interpretations of quantum theory.
Well, that's it for this edition of Light Matters. For more on any of these stories, visit Photonics.com. To subscribe to our newscast, you can click on the share icon on this video player and choose feeds to enter your subscription directly into iTunes or any other RSS program.
There are also icons to share this video on most social media sites, and you'll find Light Matters in the iTunes Store and on YouTube.
Please send your questions or comments to lightmatters@photonics.com. We'd love to hear from you and maybe use your questions on a future show. You can also find Photonics Media on Facebook and Twitter. Thanks for watching, and remember, if it's light, it matters.